floppy. We saw one last year.
Well, actually, they used to be. When I was in my second grade computer class, we had an actual floppy disk, and then I never saw another one again.
I thought these were from like the 50s.
Nope.
The 50s?
Computers,
Yeah.
home computers don't even exist till about the late 80s. Okay,
Huh.
so we have the compact disc. Now, let's say you have some music on your computer, and it's very important to you that you burn a CD for someone you've just met and are very interested in. It's important they understand how you feel about them, give it to them.
Did you really ask?
Like a mixtape and
Did
a
you really
mixed
think that
CD.
we wouldn't know what a CD was?
That's so stupid.
They still have
You
Taylor
burn
Swift.
a CD for this person. Now here's the thing. Your computer is the nucleus. It has the code inside it. Does that make sense to everybody? But here's the thing. This person only has a CD player, so you can't give them MP3s or however else you share music. I don't know. Don't tell me. I don't care. So what you're going to have to do is you're going to have to carry the information to that CD player. So what are you going to make?
Copy.
A CD, right? The CD is the mRNA. It's the messenger, okay? Now you're going to translate it in the CD player. When you turn on the CD player with the CD on there and that Taylor Swift song begins, what are you going to hear?
Vomit.
Translation.
That's right, you'll hear music, right? Because the meaning of the code was music. So what's the meaning of the code in this process? What's the code?
Protein.
Protein. So what will we do in translation?
Make protein.
We'll make protein. So we'll turn what? What do we start with here? mRNA, and we'll turn it into protein. That's right. We will use mRNA to make protein. Elizabeth?
When does tRNA and rRNA come in?
Well, if you'll look, the next thing we're going to talk about is tRNA. Okay. So, translation is getting the meaning out of a code, and the meaning of this code is protein. So we'll be using mRNA code to make a protein. Does that make sense to everybody? Okay. Now, where will this take place? What is our
Ribosome.
CD player? Right, the ribosome. And of course, what's the ribosome made out of?
Proteins.
No, it's going to make proteins. It's going to be there. What's it made out of?
Enzymes.
Fat.
DNA.
Okay, so let's flip back to our list of RNAs. What three types of RNA do we have?
Transport.
Amino acids.
rRNA. rRNA. What does the R stand for in R?
Globular.
RNA. Okay.
Wait, you said the ribosomes are the CD player?
Yes, in our little metaphor, right? They're not actually a CD player.
Oh, yeah.
If you put that on the test, you'll be absolutely wrong, okay? They're like, we can think of them in that metaphor as the CD player. They're going to make the meaning, in other words.
What was the CD?
The mRNA,
Okay.
right? It's the messenger, right? That's what we can carry from the nucleus of the computer to the ribosome, the CD player, okay? And we want music, that's protein, okay? So, the ribosome is going to be made up of two pieces. And I'm going to draw this, and we're actually going to watch a little video. Hopefully we'll have time to watch the fun video. I don't know. What it's called. Huh?
Of the hippies?
Well, if we have time to watch the hippies, it actually takes a little while, so we may not have time today. But we will watch it at some point if we have time. Okay, so we have two globs, right? Remember we talked about ribosomes are globular? We have a big one and a little one, and if you watch the hippie one, you'll get to hear hippies sing about it. It's very bizarre. Okay, we have two ribosomes, all right? And these are going to be where proteins are made. And what we're going to have is our long line of mRNA, single-stranded. If you notice, how did I space them?
Threes.
In threes, right? I spaced them in threes. And how do I know this is AUG here?
Because that's what it starts with.
That's the start one. And we're someone give me another three.
T
Nope,
A U
no T's. We're in RNA.
UAC.
And one more.
CGA.
It has to start with you.
It has to do the stop, right?
We're not going to finish. It's gonna. Okay. We're not going to finish this one. It's going to go on off the board,
Why
right?
does that
Yeah.
start you?
Why does it start with AUG?
That's
That's our start codon.
Always.
Okay, so if you look back in your notes, you'll see that we start with AUG, which is our start codon. Right? And then we'll end with either UGA or one of the other U's. I don't
The
know.
one in homework didn't start
Okay.
with UAG or AUG.
You were looking at DNA, right?
Gotcha.
And it was just a random sequence of DNA.
This is always RNA.
So RNA, the first codon that will be coded, it actually has something in front of it, but we're not going to worry about that. The first one that gets read is this, okay? You can think of it, there's stuff in front of it, that's like the pages of a book. Like you don't, like it says copyright and all that stuff, you don't read that. This is the first word of the book. Does that make sense? Okay, so we are going to run this thing through here, like we were pulling one of those little helicopters that you rip cord and it zooms off. This is gonna pull right through there, but nothing's gonna fly, nothing's gonna fly.
Those fairy things.
Yes, sure. Okay, so these are going to move through our ribosome, okay? And I'm going to erase this just to give myself some space here.
into the fire.
Okay, mRNA, ribosome.
That's ribosome?
Ribosome,
Okay.
mRNA,
Got it.
okay? Now,
They're
once
both
we
ribosomes.
get to this point, the ribosome itself, and I don't know, well, yeah, you don't need to know that much of CD players. Anyone know how a CD player works, by the way?
Yes.
You put it in the.
No, okay, that's not enough. It shines a laser on the disc to read it.
Oh, it's the
okay?
rainbow.
That sort of.
Yeah.
It shines a rainbow on there. So the laser is what actually reads the CD, okay? So the ribosome itself is just where this happens. It doesn't actually read the information. Instead, and I'm going to move our strand over here.
Look a sandwich.
It rips.
Are you hungry?
And then I forget we put here, so I'll make one up. Okay. So instead, we need something else to read this. But in addition to reading it, it has to bring us our supplies. What supplies do we need to make a protein? What are proteins made out of?
Amino acids.
Who's going to carry the amino acid?
mRNA.
Transport.
Transport RNA. tRNA is going to bring in our amino acids, okay? Now, how does it know which amino acid to bring? If you'll remember, each tRNA, I'm going to draw it kind of like this, has 3 letters sticking out right here. Everybody remember that from our drawing?
Mhm.
3 letters sticking out. Now, if the letters C, C, C are here, is it going to stick right here?
No.
What's the only tRNA that'll stick right here?
U. Wait.
UAC.
It's going to be U, A, C. Right? I know I didn't really fit right there, but. Okay, does that make sense to everybody? Now, we happen to know who is always stuck to UAC. Which
AUG.
amino acid does AUG code for?
I got this.
A.
Stop.
U. G.
Start. My bad.
Methionine. In fact,
Yes.
right here,
Methionine.
I'm going to have methionine. Does that make sense to everybody?
Okay, what is that gonna do?
Okay, tRNA is a truck, right? And if the truck has the tag UAC, what will it always be carrying?
AUG.
Well, no, that's what it'll match with.
Methionine.
It'll carry methionine. Okay.
To match with
Okay. So AUG is on the mRNA, right?
Yeah.
This is the mRNA. This is the tRNA, okay? It needs something to match the codon. What are we going to call the thing that matches the codon? In our notes, perhaps?
Anticodon.
The anticodon. Just like the mRNA has a bunch of codons, each tRNA has one anticodon that matches one and only one codon, okay? And each tRNA matches one and only one codon. Isabel?
Wait, what did you say? Anticodon?
An anticodon is 3 nucleotides that match a codon. And so it's on the tRNA. Each tRNA has an anticodon, each of which fits onto one mRNA codon. So, what type of anticodon will match right here?
A. G. C.
A, G, C. Now, I don't know what that carries off the top of my head, but how can we find out?
A.
Serine.
No. What are we going to look up?
The chart. AGC.
Chart.
Serine.
Okay, hold on 1 second. Are we going to look up tRNA or mRNA? That chart is an mRNA chart, so we're going to look up the mRNA, okay?
So serine.
How do we know which one?
Yeah, it's both.
You
both-
only have
it's
one
the same
chart.
thing.
You only have one chart. It's an mRNA chart,
They're
okay?
both serines.
Good. But we always look up the mRNA on an mRNA chart. Does that make sense to everybody?
Wait, which one's we have? The small one.
So the long strand is mRNA. The
That-
one that's kind of shaped like a T is tRNA. Quiet.
I thought at this moment though the one on the bottom is the mRNA, it's the messenger one, right? And then the one up top that has the 3 prongs, isn't that tRNA?
Uh-huh.
So they're both RNA right now.
That's right, but the chart you have is an mRNA chart.
Oh, an mRNA. Gotcha. I thought you
So
just
the
said
chart you have, in fact, I don't think I wrote on that. If you want to write above that mRNA, keep in mind that's the mRNA chart.
So what does the anticodon come in to do? Like it comes
Okay.
in.
So each tRNA has a different amino acid. It doesn't know where to line up, except that its anticodon will match part of this code. So if I want serine next, I will call in the serine tRNA by having this sign happen next. This word says serine because it matches up with a tRNA that carries serine. Okay? Once they're both here, I'm going to move over one, and when I do, I'm going to break this bond. But when I break that bond, what bond will form? What am I trying to do here? Make what? And proteins are made up of what things stuck together? So what will this bond to?
The other one.
Right here. Okay. What if my next one is G, G, G? Who's going to come in?
CCC.
Okay, so tell me, what amino acid comes in from CCC?
Proline. No, just kidding.
Yeah, proline.
Sorry.
Okay. So it's going to come in, and by the way, this all moves over. And then when it moves over again, this bond's going to break, and what's going to happen?
It's gonna shoot off.
And what are we going to make bit by bit?
A square
DNA.
ribosome.
We're not going to square it. We're just going to add one after another after another. Or a long chain, and that's what amino acid is. I mean, that's what protein is. A chain of amino acids. Shh, quiet. Yes, Amy?
Sorry, I just was making.
Okay. Any questions on that? We're going to watch a little animation.
So
of
an
that.
amino acid is the chain of proteins.
An
That's
amino
what
acid,
that
no,
is.
a protein is a chain of amino acids.
It's the chain that makes us.
That's a chain of amino acids is a protein. Alright, let me just call this
How
up.
many is considered a chain?
There are no proteins that are really shorter than about 100, and most of them are a couple thousand. Right, but I'm not going to draw
Yeah, no, I know, I know.
1,500 of them,
I
right?
know, but just the more protein you have, the more healthy you are.
Well, so the how long they are will depend. Tell you what kind of shape they can form, which is what tells you what they can do. But it's not like more or less or better or worse.
Well, wouldn't it matter with food?
So the longer they are, the more amino acids they take. So that can relate to it. Okay. So if we watch right here, and it's gonna cycle around in a second. This one I don't know. I just found it somewhere, but I'll show you a website you can look at in a second.
Is it
Okay, so
Turk.org?
what are these two brown blobs?
Ribosomes.
Those are ribosomes. Those are ribosomes. What's my long strand with all the letters?
mRNA.
That's mRNA. What are these weird yellow squigglies with three
tRNA.
things? tRNA. What are the little pills up here?
Amino acids.
Amino acids. Okay, so it's going to start again in just a second. Okay. My first one, if you notice, is going to say AUG, right? And it's going to, come on, recycle. AUG. So what do we start with?
Whatever.
Methionine. Next one comes in, that's
Valley.
valine. Then this is going to move over one space, and we're going to have someone who matches CGG come in. And who's that?
GCC.
And that gives us
Arginine.
arginine. And then they form. And this is going to continue on until we reach what?
UGA. Or
Usually one of those stop codons.
Wait, where is it? Where are they going?
Who?
The.
Where does the tRNA go
trucks?
after?
Okay, so let's say a truck comes to the work site and unloads all the lumber it carried. What's it going to go do?
Back to get
Deliver.
more.
Pick up more lumber and come back. Right?
Wait, it's delivering?
So, okay, so this is where we're building. So it's delivering amino acids.
Right.
So once it's delivered amino acid, it's going to go back, pick up another amino acid, and come back.
Okay. No, each.
What happens if there's not a multiple of 3?
Well, then nothing will form there. You can't make a
It's a
protein.
little bomb.
So let's say if we knocked out this G and there's nothing here, then we couldn't make it. It would stop.
Okay.
Margaret, and then we're going to skip ahead. You're going to see it stopping.
What is-
Up there where you have methine or whatever.
Methionine,
What's the one next to it has?
serine. But we just, it's not, it doesn't have to be. We just made that up because I wrote
Those are
UC.
the letters you use. And then the pro is the.
Proline was CCC.
Okay.
And
And it
what,
goes
this is the only one that is always the same. We just made these. We just picked them randomly.
What's the?
I have 2 questions. Do these have anything to do with transport proteins?
Okay, so this transport proteins, no, those are made this way, but transport RNA and transport proteins do not have anything especially
Okay,
connected.
and this is happening in every cell?
This so this is happening in you probably about at least I don't know 5 to 10 trillion times, right? Every instant.
Wait, so it is every cell?
Every cell that's making a protein. So your fat cells aren't doing this, or if they're doing it at a much slower rate, but your brain cells are doing this constantly. In fact, they're doing it faster than anything else, and then your liver's doing it unbelievably fast, especially if you just ate lunch or if you just ate snack or whatever. Your liver's going like crazy doing this.
What's the release factor, and why does
You
it
notice
make
it's
the
not
ribosome?
tRNA, right?
Yeah,
So the release factor stops the whole process.
and it breaks the ribosome.
That's right, because we don't need it now, right? We're done.
Oh.
Now, and it's UGA. Hit stop codon. Now, we'll bring the ribosome back together for the next protein we need, but we're done with this right now. Does that make sense? Yes, ma'am.
Does it always hold just two, like the T and the A?
This is not important, you don't need to know it. There's a little disagreement about whether there's 2 sites or 3 sites, right? This is called the peptidacyl site. This is the aminoacyl site. This is the first binding site. There may be what's called an E site over here, but it's not. But for us, there's just 2 spots.
What's the difference between the two? Like, why are there two?
So this is the spot we release the tRNA from, and this is where the tRNA attaches. So it comes in, goes out there.
Where does the release factor come from?
It's floating around. It's something you make.
What is it?
I believe the release factor is a it's a tRNA, but it has an ion in it, and so it stops the whole process.
Do we have
I
to
think.
know?
Nope. I'm not even certain about that. I will say this: it's almost always colored pink. I've never figured that out why.
Like a bladder.
Right. You talking about human bladder? They're not that pink.
What color are they?
They're tissuey color. They're kind of pale with red streaks where the blood vessels are.
I thought they were pink.
Yeah.
There's almost
cartoons,
nothing pink in the natural world, right?
Your brain.
Nope. Brain's gray. Heart is like dirt red.
No, I saw a brain before.
So the only pink comes from shrimp, and so the flamingos, in order to be pink, have to eat a lot of shrimp to turn themselves pink.
If we eat a lot of shrimp, can we turn pink?
Only one way to find out. Okay.
Eat a lot of
But
shrimp.
I don't like
So
shrimp.
let's take a look
I'll do it.
I only like boiled.
at this. Okay, so if you remember the site we looked at for mitosis, John Kirk, he also does a very, very, this is the most in-depth of all the animations. So we're going to go through it real quick, okay? What's this long strand going to be?
mRNA.
What are these?
tRNA.
Ribosomes. What's this?
tRNA.
tRNA. Now over here we have, if you notice they're not very big, all the amino acids. Everybody see the amino acids? Now what's interesting is he actually goes in and shows you the form. If you notice, adenine and guanine have how many rings?
3.
2, right? And how many uracil and cytosine have?
3.
1 ring.
I don't know what I'm saying anymore.
Okay, so you see, this is a ring of atoms right here, right?
I don't even know what a ring is by now. What?
And that's another. Okay, so
Oh,
you
the
see-
hole, the little yellow hole.
Quiet. Do you see the light blue space here, where the finger is on?
Yes.
Okay. Do you see the light blue space here?
Yes.
So that was 2, right?
Yeah.
How many do we have here?
Just 1.
Okay, so that's a ring around the empty spot, and that's 2 rings around the empty spot. Okay.
Go ahead now.
So, what's going to happen is, if you notice, there are 3 things sticking out here. What's that going to be?
The thing.
What do we call the 3?
Codons.
Okay, these are codons because they're on tRNA. What do we call it when it's on tRNA? These are anticodons. They're going to match up with
Codons.
codons. Okay, so here's what's going to happen. First, we attach the amino acid. By the way, what amino acid is that?
Methylene.
Methionine, because we're starting. This is all the other tRNAs. Then our ribosomes come in. See the 2 spots? It attaches. What does it attach to?
The AUG.
AUG.
Question, is the left side a close-up of what's happening?
That's a close-up of, see the big star right there? And that's the circle of what's happening right here.
Okay.
So this right here is a close-up of that. In fact, as it goes down, we'll see it move along. So whatever comes in next, we'll see. So that's 3 A's. What does it bind to? U, and that makes. By the way, this, I think it's not doing it yet. Once you're done with this, you can run through the chart and I'll tell you exactly what everything is. So, then they bond together. That comes together, they bond together. Comes together, they bond together. If you notice, this is getting longer and longer and longer.
It's like a game.
I suppose so. Or just a process.
Just.
I like it.
So, and eventually,
It
see
does
the
look
pink
like
thing,
it.
what's that going to be?
The release.
The release factor. That's right, because UAA says stop. There's no anticodon for that. And so that's going to get stuck in here, and if you notice it wiggles around a little bit. I have no idea if it really does that or not.
It's like a wrench.
And then
And so
it breaks it apart.
That's the protein right there.
This right here is the protein.
Okay.
Dang.
And the invention of this
Try
protein
to
has only just
name
begun.
can be tried to make as big as a protein for the
Sure, sure. Okay, so if you notice,
Yeah, I know.
this process
I'm
happens,
stressing out about
and we form our protein. This is a very, very short protein. It's not even really a protein. It's actually a oligoamino acid, but you're right. So it just wants to show you what happens, though. Yes.
Oh, what was the other one we watched? Was there a website?
I found it somewhere. It's an animation I found. But John Kirk, if you want, there's tons and tons of animations of this, though, if you do want to look them up. But the John Kirk one's very accurate and easy to follow.
That's easy to follow.
Compared to some of the other ones. I
PowerPoint.
think, let's see, how much time do we have?
Oh, can you share? Is there a way to
15
share that on the
minutes.
Google Classroom? The share
Probably. I just gotta figure out how to do it.
Can you show us the funny one?
Hold on, I'm seeing. What time does the class get out?
35.
Okay.
Alright.
The next slide was dance, and you didn't show us that one.
No, wait, I clicked the wrong thing.
Dance.
Did you do that?
I don't know who's.
You gotta do what?
I thought my pencils
I
were
mean,
off, so I
made
just
this
ignored it.
Oh, wait, you said you just
I
found
kind of
it
got
somewhere.
into.
She asked about the animation,
Little speaker.
right?
But he put it together,
Uh-huh.
Oh.
right?
Why does it say dance?
You're about to find out. Alright, if you get the Windows. To explain this real quickly, because it needs at least a tiny bit of explanation. People at Caltech, one of the leading scientific research universities in this country, were asked by educators to speak to the youth of today. And this is a bit like a well-meaning teacher today deciding to wrap their lesson so it can relate to their kids. Instead, this was done in about 1970, and so cool young culture at the time was the hippie movement. So this is what they came up with. I will say this, it's unbelievably accurate for what actually happens. It's also hard to get over the fact of what these people are doing. And the narrator's really bizarre.
I cannot read this.
Oh, that's just much.
was brilliant in the slidey 30S ribosome with a gyre and a gimbal in the wave.
So that's a ribosome.
30S ribosome.
Why are they just glomping all over the place?
They're touching. It'd be like Mr. Teal's class with the.
Our Mindy was in on it. That colored message letter ran.
Now, how many people are together?
30.
And if you'll notice, each balloon is a different nucleotide. You have a different color balloon for each nucleotide.
On your head.
You like
This is so
this?
creepy.
Yeah.
It reminds me of the Illuminati.
Okay, so you see how the single strand? That's mRNA and codon.
What are these children doing over there?
That's a ribosome.
Oh. Obviously. How could I miss it?
I'm sorry.
The music is what.
I love it.
The music is what.
I want to know what the song's called so I can buy it on iTunes.
Wound they were in the glorious sun by initiator factor 1.
Is it better?
Oh, I should warn you, the amino acids are guys basically in diapers. I don't know why.
Oh, okay.
Who's the initiator factor? AUG?
So this actually goes into more detail than we use, right? So we're not. It's basically a coenzyme.
That's just what
Why
works
doesn't she
together.
have real clothes on?
So they just make
Initiator
it
factor 2.
Did initiate-
Initiator factor 2.
Did initiator factor one make methionine?
No.
But that would
It's a
be
part of the process.
since you're the ballerina. What would that
Why does it
be?
seem like we don't have the detail?
There we go.
We need
Alright, see
to.
the amino acid? At least he's got shorts on.
Oh,
Oh, there he is.
that poor little pet kid is all left out.
Okay, every time they blast the smoke, that's ATP releasing energy.
Wow, they were really high
I
tech
know,
for their
I know,
day.
They do it. It's actually a fantastic representation of what's happening. And what's interesting is that even molecules, they actually probably move pretty similar to the way these people are rolling around. That's how bizarre this is.
How did they get this on the internet?
They probably aced the test.
No, it's an old video they put on the internet.
The 50S ribosome.
They didn't put it on the internet back then.
How are they not dizzy?
So there's the other part of the ribosome.
Oh yeah.
She's singing a ball.
These guys are creepy.
Do they know what they're doing is not normal?
They don't care.
Been smoking the weed.
That's what it's from.
They're not smoking weed.
Hey, they're bringing an S.
California, right?
Caltech, Berkeley.
Wait, is it a?
Berkeley, that's in.
Wait, is it high school or college?
No, yes, not even
Just watch, just
Gathered
watch.
in tRNA.
So once again, amino acid top is Berkeley.
I want a poncho.
What's that one guy?
He's the amino acid. All the amino acids, for reasons I don't understand, our guy's not sure.
Came glumping back.
Glumping?
But they also try to make it like the Jabberwocky by Lewis Carroll. I don't know why.
I don't know.
So they form hydrogen bonds. You see those hydrogen bonds?
No.
Yeah. it's-
Yeah.
The people on the hand.
Look at him.
Amino acids both inside a lab.
Uh-oh.
Peptide bond was formed.
Being kidnapped.
So, 2 amino acids now together.
And as the
Peptide bond.
the messages shift.
There it is.
Oh, there goes. It's going through.
Yeah?
We need a name for that.
It's called indexing.
Indexing.
Even the football players are getting in on it.
We need a better name for it.
tRNA.
Put it on the drugs.
What are all the other people floating around outside here?
They're actually, because this happens in a cell, there are 3 molecules floating all over the place. So, index and
Let's do that. We need to make our own video. We just
They're so happy.
all the parts and then put them together.
Can we do this?
Yes, we don't have enough
there's amino
people.
acid prancing in.
Wait, is that a boy or a girl?
tRNA approached the site, bearing its amino acid load. The sequence was determined by the mRNA message unit magic code.
Magic code?
Okay, so.
No, it's not
You
over!
guys see that. Once again, it's actually very accurate. Of course.
50, what do you say, 50S ribosome?
50S
ribosome.
50S!
How did they like add in music and stuff?
Because we are done
We're done with class?
with this.
Oh.
Okay, so while I can't find the remote, I can still do this. Alright, so now we want to take a look at what can go wrong
So she can see
with
it.
this whole
The
process.
video.
This whole process of
I
creating
figured she went to see the Happy
our
Dancers.
amino acids.
Busy's head.
What can happen when this messes up?
Dang it, I missed it.
Mutation.
Dang
Every
it.
time we're in this class, you take 1,000
Okay,
pictures
so
of Busy.
over
Well, this-
time,
that's
quiet. This code, like any code, can begin to be improperly written. Mistakes appear. Anytime you copy something enough times, imperfections are going to occur. And actually, if you take a piece of paper or with writing on it, if you take a worksheet or notes or whatever, and you copy it once, and then you copy that copy, and you copy that copy again, eventually, surprisingly, it'll be completely illegible, right? It'll accrue, it'll build up all these different imperfections. The same thing can happen as this process happens, as we copy our DNA. And these processes can make changes, in the genetic code, right? We can actually have changes in the genetic code. Now, we call these changes what?
Mutations.
Mutations. Most mutations are actually harmful. In fact, the vast majority of mutations are harmful. So when the mutations happen in you, and there's probably at least a couple happening to you today, maybe some happening right now, your cells will notice the mutation and they'll kill the mutant cell, right? Or the mutant cell itself won't be able to survive because of that mutation.
Okay, so we're talking about cell mutations. It makes me want to ask about like X-Men.
Okay, so no, they're not real.
No, but could that ever happen?
Wow.
A gene mutation that like
makes you shoot laser
I don't know
eyes.
what X-Men
No.
is.
Okay, so genes code for proteins. So you would need a laser producing protein.
We can make that.
But could you have one that like animals have
Well, you'll
wings?
use proteins to make those lasers.
Animals have some animals have wings. What if we had those sort of proteins and a genetic
You
mutation
couldn't
made us
fly.
grow wings?
Your body's not built for flying, right? If you wanted to fly, you'd need wings, and then you'd need legs like a bird, and a body like a bird, and a heart like a bird, and lungs like a bird, at which point you'd be a bird, right? So if you wanted to become enough like a bird to fly, you'd be a bird, okay? So the X-Men who has wings on his back, in order to just wave those wings, he would need entirely new muscles that were larger than all his leg muscles combined.
Do
Right.
people try to do that? Like,
No.
think they're birds?
Uh,
I mean, I kind of can see that happening.
well, some people jump to their deaths if that's your ass.
Like that guy in the
Why-
mutation.
Some people just
Why? Huh?
Why do mutated cells do?
Well, so you have, in your body, you have a whole group, a whole gang of cells called, they're actually awesomely named natural killer cells. And their job is to blow up your cells who've gone bad. And that's their entire job. They travel on your body, check on cells, literally rouse them up and hey, what's wrong with you? And they check all their stuff. And if they're dangerous, they just kill those cells immediately. That's their entire purpose, is to kill your cells that go bad. They're called natural killers.
And so
If I was a
they
cell,
killed
I'd be
the
dead.
mutated ones.
If they find a mutated cell that seems dangerous, they'll kill it. Okay? Now, while most mutations are harmful, some are harmless, some don't seem to have any big impact, and occasionally, only very rarely, mutations are beneficial. I'll give you a quick example of when a mutation can be beneficial. Humans have genes for dark pigments. But if those genes mutate, they don't form, and you can have blue eyes. Blue eyes are very beneficial if you live not here, but if you live somewhere where you find blue-eyed people. Where do blue-eyed people?
Sweden.
Northern Europe, right? Northern Europe, even the northern part of Eurasia, of the upper Russian areas. Because there's so little light there, you want light eyes. Dark eyes absorb light, so you can't see as much light. But they act as sunglasses. People with dark eyes can actually withstand more light than people with pale eyes. So having light eyes is advantageous if you don't live in Alabama, right? If you live up north in Canada or somewhere where there's not much light, you can actually see better during that time when there's not much light.
Is that why? Well, I don't want to sound like
Too late.
me. Well, is that why some blue-eyed people here have to wear glasses more than others?
Right. So
Because I know
if you have
my
light
sister-in-law.
eyes, if you have light eyes over a lifespan here, if you don't wear sunglasses all the time, if you have very pale eyes, you're much more likely to have eye problems. However, if you live up in Sweden where there's not much light and you have very dark eyes, you oftentimes have a problem seeing, especially in winter, right? You actually can't see as much. Probably the most interesting mutation, and one we don't think of as mutation, is the fact that unlike all other mammals can only drink milk when they're babies. In fact, humans have a gene that turns off their ability to digest milk. However, almost all of us are mutants because that gene broke. And what can we still do?
drink milk.
Drink milk. And what do we call the normal people?
Lactose intolerant.
Lactose intolerants are the non-mutants. It's the people who can drink milk that are actually mutants. And that mutation actually is beneficial because if you can drink milk, you have an extra source of energy that people who can't drink milk. Now, it doesn't matter to us now. We have an abundance of food. But if you lived, let's say, on a farm and you grew crops and had a cow and you could only eat the crops and someone else could eat the crops and drink milk, they're going to have a longer life. They're going to be more likely to survive because they have more energy. Amy?
So, if you
Yes, but not as bad as it would be if you had light eyes, right? So sunglasses are better, and by the way, as you age, your eyes will get weaker and more vulnerable to light, which is why, whereas younger people will fashionably wear sunglasses, older people need them more. I'll just give you an example now. I never wore sunglasses at all because the people I knew wore them all the time were always tools, so I was like, I don't want to be like these people. Now I cannot go outside without sunglasses. Like I can't drive a car because my eyes have weakened because I'm old man eyes.
You are barely 30.
I did. All right, our homework tonight. We got lots to cover tomorrow. Page 301 and 307. Even though we may not have gotten far enough to do 307, just read the chapter and answer it. So 301 and 307, that's your homework. Just to read. So if we haven't gotten to 307, you might need to read the chapter. I'm not sure if we got far enough. But answer any questions.
Is it 1 through 5?
You're going to answer 1 through 5 on both these pages. If we didn't go over those, read the chapter.
Go read the chapter.
Yes, ma'am.
Remember, like, sections go